Professor Han, for the kind introduction. And it's a great pleasure to have the opportunity to present at ICANX. And also, it's wonderful to meet our old colleagues virtually, and looking forward to our in-person meeting in the future. And also, thank you everyone for coming to the talks. It is a great pleasure to interact with the world-class experts and the researchers here. So our research group it's located at the Department of Applied Physical Sciences at UNC Chapel Hill. So our lab is dedicated to developing innovative devices for advanced healthcare. And one of our core objects is the design of bioelectronics as the key to achieve precision medicine. So maybe the first question you might ask is why precision is so critical in medicine. So we see recent breakthroughs have made electronics adopt soft mechanics, which gave them adaptive freedom in space to conformly interface with living tissue. So being adaptive is not enough, as most biological system is dynamically evolving. So to truly realize a seamless integration with the biological tissues, the next generation bioelectronic devices needs to be adaptive not only in space, but also in time. So that also drives expanding research efforts in precision medicine. So however, the challenges which also frustrate the medical community is the mismatch as a abiotic interface. For example, when a rigid electrode is inserted in a human brain tissues, the tissue damage and scar formation may happen due to the much higher mechanics from the electrodes that inserted into two orders softer brain tissues. And the second is when a metallic surface or even a biocompatibly coated surface of implants contacts with the biological tissues, so the corrosion may appear, which even though it takes years, maybe decades, and those corrosions, even though it's very small amounts, could disrupt neuron functions and normal metabolisms. So in this presentation, we will introduce three cases of materials processing and the device developments towards precision medicines. So the first is a Kirigami inspired 3D microfabrication technologies. Especially focus on bending monocrystalline silicon in 3 dimensions. The second one, we will show the autonomous drug delivery system, which could improve the precision in the pharmaceutical administration. So lastly, we will talk about a wearable technology for muscle tracking that can leverage the AI algorithms to improve the precision as well as the long-term variability of the device. So the first one is the 3D folding of monocrystal silicon. So the microfiber folding strategies introduced here. So starts by integrating a 2D precursor. In this case, is a monocrystalline silicon nanomembrane. So with a folding host, which could be a soft substrate like a silicon, through a customized transfer printing process. So strategically bending the host of the substrate at various angle translate the origami effects to the guides to the precursors into a specially engineered 3D microstructures. So such a host guest strategy can resemble such lever mechanisms and precisely alter the structural reconfigurations through the folding registration and folding angle to navigate the folding trajectory of the micro-scale guest towards a more complicated 3D structures with a higher precision of manipulations. So, and the results is turned out into a 3D complex structures of monocrystalline silicon, which is widely used in the semiconductor industry, that is anchored on the edge of a substrate instead of on the planar surface of the materials. And also, the simulation confirms the maximum strain in the folded silicon well below the fracture threshold, which indicates the stability of such a highly folded, highly strained structures. Even for the silicon, which is generally brittle materials compared with other broad range of materials collection. So here we demonstrate the applicability of microfolding strategies to monocrystal silicon nanoribbon, which is intrinsically brittle, and the silicon folding process is highly reproducible and robust. And from the collected results, we could observe a flat top in all those highly folded silicon nanoribbon structures, so which has not been reviewed in all the previous reported silicon kirigami, probably due to the high strain of silicon folding as well as the customized transfer printing process. So not only the nanoribbon form of silicon, so the micro folding strategy can also use a more complicated 3D 2D precursors of silicon. So here we shows double helix structures, and also we can use the finite element analysis to confirm the strain distribution inside the silicon, and also summarize a scaling law to predict the maximum strain in silicon as a function of the design parameters. So the microfolding approach allows the 3D folded structures situated not only at a planar substrate, like the traditional 3D microfabrications, but also it allows the structure to sit on the edge or a cylindrical base, which enables the anchoring of folded robots inside casters. So such integration with a caster allows the robots to be fully enclosed inside the miniaturized tube, which features a minimally invasive implantation to minimize the local tissue damage. And then when the caster reach to the target tissue surface, so in this case, could be a heart, the folded structure can bloom and expand from the caster to establish a more conformal interface, even with the miniaturized caster tubes to more comprehensively measure the cardiac tissues. So here we could embed so 4 strain sensors into the Wearable robots, and the conformal interface of those strain sensor allow us to quantify the local cardiac contraction continuously and in real time, so which is valuable for diagnostic information for diagnostic cardiac surgeries. So here we use a mouse model and deploy a Wearable robots onto the mouse heart. So we see the 4 strain sensors can precisely monitor the contractility of the 4 respective chambers of the heart, and differentiate the abnormality of the local cardiac tissue damage from the myocardial ischemia. So then we move on our second development case, which is drug delivery devices that's to hope addressing the issue of drug overdose and drug use disorders. So as we know, in the US alone, so drug overdose and addiction contributes to nearly 75,000 deaths annually. So making these issues one of the leading causes of mortality among working age populations. So one reason to lead this is the manual operation of the drug administration, which often misses the optimal window and sometimes cannot be achieved the needed precision in the dosage as well. So another user case for the digitalized drug delivery platform is the insulin delivery for patients with diabetes. So although there's an electronically controlled mechanism on the market, like the insulin pump shown here, for the delivery. So the pump device, as you see, is still bulky, and the needle is designed into a long, thicker shape, which presumably can generate tissue damage, especially during the daily activities. So we developed digitally automated drug delivery devices. So the concept is to build a digital switch on each drug releasing reservoir. So in this case, it's a microneedle to allow the microcontrol chip directly control the drug release. So following the established work from Professor Robert Langer, we designed the digital switch using those deposited nanomembrane of a goat, which can act as a barrier to prevent the diffusion of the drugs during the standby stage. Then, with a small electrical trigger sent from the microcontrollers, the gold nanomembranes can undergo electrochemical corrosion to initiate the drug release. So from this strategy, we could observe the dense gold coating nanomembranes can effectively block the diffusion of the drugs, and then initiate a very observable disintegration of the coating within 30 seconds, which is a relatively short time, and to expose the drugs hidden underneath in order to initiate the drug release. So one advantage of using microneedles at the drug reservoir is to achieve high spatial controllabilities. So here we show each using a beam of laser, we can draw the circuits onto the gold-coated membranes that is situated on top of the microneedles to allow a digital switch on each microneedle to be individually controlled by the microcontroller chips. So therefore, we can manipulate both the time and the location of the drug releases from the microneedle arrays to some extent. So this would allow us to create a small wearable patch with potentially hundreds of drug reservoirs or array of drug-loaded microneedles. So all controlled wirelessly via the Bluetooth microcontroller. So this enables the precise timing and the dosage of drug release, potentially in response to the health monitor data to form a closed-loop system for optimized drug deliveries. So we validated the device efficacy through a mouse model with the brain implantations. The immunohistochemistry data reveals a good biocompatibilities, which is consistent with other commercialized brain implants to demonstrate the minimally invasive microneedles suitable for implantations. So we can also provide electrical stimulation with a gold-coated layer for voltage that is smaller than the gold working functions in order to serve both functions, the electrical stimulation as well as the drug deliveries. So the animal studies results demonstrate the effective encapsulation with the gold, as well as the observable drug release so as soon as the gold protection layer is triggered and disintegrated in the brain tissue environment. So those results could serve as a proof of concept for a more larger scale animal study using such digitally controlled microneedle drug delivery systems. So then you might question the broad applicability of such gold encapsulation process. So as the deposition of the gold requires a vacuum environment, as well as the elevated temperature close to 100 degrees Celsius, so which may damage so many drugs, especially those genetic drugs, which cannot survive at a slightly higher temperature. So to resolve this issue, we use those solution processable approach to allow uniform coating of metallic materials, which in this case is the 2D metallic mixing. And still under a voltage bias, 2 to 3 volts in the biofluid environments. So the mixing can undergo redox reactions, and these reaction can enable local volume expansion, and that expansion can fracture the encapsulation layers to expose the drug protected underneath. So to enable the controlled drug release. So the mixing, as well as its oxidized byproducts, are both highly biocompatible. So as validated by many literatures online. So the third device we will introduce is aiming to leverage the AI algorithms for wearable sensing. So especially focusing on the muscular therapy and haptics. So as we know, the continuous muscle tracking is vital for most muscle recovery as well as the haptic technologies. So it opens the door for future generation haptic system to function as seamless as our own body parts, and also aim to make the rehabilitation more efficient and as well as affordable and effective. So, however, the current muscle tracking technologies on the market face several limitations. So the EMG is often plagued by excessive noise, so prevents its capability to quantify the muscle movements. And then we see the ultrasound, even though it's a precise to provide imagery information, but it often requires the complex device design as well as a specialized attachment layers, which typically a hydrogel and that can degrade over time after a few hours, maybe a couple of days. And then also it generally requires the user to be in a static structures. Also, this limitation prevents such device to enable a very long term of wearing capabilities. So to address this issue, we utilize the near-infrared light, which is not only safe and has sufficient tissue penetration depth, but also demonstrate a very selective absorption by muscles. So that means so the muscle local reconstruction or movement can generate a corresponding backscatter light, which contains the muscle movement information that enable us to analyze and monitors. So then we can carry out a numerical simulation to confirm such correlations and its effectiveness using the near-infrared light to track a very subtle muscle movement with the illuminated tissues. So you might wonder if the light is so effective, why there's no suitable commercial products on the market. So I think the primary challenge is the non-linear correlation between the light signal intensity as well as the muscle contractilities, which we think the machine learning could play its role in order to generate a meaningful quantification in the signal to physiological parameter correlations. So we designed a wearable patch incorporating 4 near-infrared sensing units along with a Bluetooth chip to wirelessly transmit the data So in addition to the near-infrared sensor, we also integrate a gyroscope to differentiate local muscle motions from the neck, as well as the overall body movement artifacts. So we use the machine learning to process those transmitted raw data in order to provide a clinical-ready biofeedbacks for clinical doctors by identifying key muscle movement for diagnostic purpose. So those real-time tracking data can be fed into AI algorithms, which could classify muscle activities and also potentially quantify its severity or muscle movement amplitude in order to track their occurrence frequency over time and assess early signs of muscle recovery or potential applications. So our measurements results using the wearable optoelectronic systems reveal well identifiable signal signatures corresponding to a specific neck muscle movement of interest, ranging from deep breaths, so swallows, a dry cough, and the throat cleaning, as well as other more overall body movements. So those signal signatures would allow the AI algorithms to carry more specialized event classification as well as quantification. So here we show a video to demonstrate the device in action. So as you see, so the a previous generation of device can be attached to the neck, currently using a catheter. Maybe in the future we can design a more ergonomic band-aid or the neck wrap. More importantly, we see as the user is undergoing awareness of neck movements, you could observe a well-defined signatures corresponding to such movements. Then, as the user changed to a different muscle movement, the signal signature changed correspondingly. So that serves as an experimental basis for the AI algorithm classifications. And then the gyroscope can differentiate the overall body orientation in order to further feed into the raw data in order to mitigate potential motion artifacts. So here, in the interest of time, we will fast forward. Then we could feed those processed raw signal into a dual model neural network to initiate the classification and potentially enable quantification on the severity or amplitude of those muscle movements. So the preliminary results on the machine learning analysis looks acceptable. So it shows a good labeling of the predicted events inconsistency with our manual classification, as you show, say, from the bottom row figures. So we also compare our wearable devices with the state-of-the-art commercial EMG sensor, showing good correspondence. As we see, the EMG data generally contains a higher degree of noise and may not easily mitigate from the motion artifacts. Also, the raw commercial device looks very bulky and not easy for long-term wear. And then we can also compare our devices with the gold standard, which is a video stroboscopy that provides a real-time video recording, but cannot be comfortable in long-term wearing. And also the results shows a good correspondence between the imagery capture as well as our data correspondence from the wearable optoelectronic devices. So we also tested the wearable patch in the real-world scenarios, which cannot be provided by the gold standard videoscopies. So the AI algorithms can score the signal patterns to distinguish targeted muscle activity from the general body movements, such as during the basketball game shown here. So the device also offers excellent comfort for long-term wear, and with the motion artifacts captured by the gyroscope. And then we can using the AI algorithm to decouple those motion artifacts in order to enable a high quality of a recording, primarily dominated by the neck muscle movement. So, and then we can use the machine learning to label the likelihood of those events over the course of the activity and analyze the occurrence frequency and amplitude for those events, such as coughing. So those information could be very useful for clinical doctors to adjust the rehabilitation schedules for diseases patients with muscle damage or athletics that requires recovery of their muscles as well. So that concludes the overview of the three inventions. So overall, our goal is to address healthcare challenges by developing novel devices. So our approach starts with materials processing, structure integration, and pushing the boundary of what current medical technology can achieve. So in closing, I would like to thank all the members of our research team, as well as our collaborators who played crucial roles in turning these ideas into realities. And finally, thank you very much for your attention.
Thank you, Wubin, for the wonderful talk.